In some previous videos, we already discussed what is glycogenolysis and what is gluconeogenesis. But both the pathway has some similarities and differences. So in these videos, we discuss what is similarities and difference in between the both pathways. So we already know about the terminology about the regulation of the glucose in the blood. So there is a glycolysis, which means breaking down glucose into the pyruvate and the lactate. Gluconeogenesis means the formation of the new glucose from the non-carbohydrate sources. Glycogenesis means making new glycogen from the glucose, and the glycogenolysis means the breaking down the glycogen into the glucose. So both the gluconeogenesis and glycogenolysis form the glucose to maintain blood glucose level during fasting. So both pathways have some similarities. So let's we discuss what is similarities between both pathways. So the similarities in the glycogenolysis and the gluconeogenesis, both pathways have to maintain glucose level in blood during fasting, and the glucose 6-phosphatase enzyme convert the glucose 6-phosphate into the glucose, and this enzyme present in the both pathways, and both pathway occurs in the liver, and glucagon activates the glycogenolysis and the gluconeogenesis. And the difference between glycogenolysis and the gluconeogenesis. Glycogenolysis is the breakdown of glycogen into the glucose, and it is a catabolic process. In catabolic process, complex molecule convert into the simple molecule. In the glycogenolysis, complex molecule is the glycogen and the simple molecule is the glucose. Whereas gluconeogenesis is the formation of new glucose from non-carbohydrate sources like lactate, glycogenic amino acid, glycerol and the propionic acid. And it is an anabolic process. Anabolic process means some simpler molecules convert into the complex molecule which is glucose in the gluconeogenesis. Glycogenolysis occurs in the short-term fasting around the 16 to 14 hours, 12 to 14 hours, whereas gluconeogenesis occurs after a stage of prolonged fasting, which is a starvation, and when the glycogen completely deplete. So in the body, after the 24 hours, there is a starvation condition when glycogen is completely depleted in the body or there is no carbohydrate source. So glucose is formed from non-carbohydrate sources like lactate, propionic acid and amino acid. Glycogenolysis has a main regulatory enzyme, glycogen phosphorylase, which break down the glycogen into the glucose. And the regulatory enzyme of the gluconeogenesis is the pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and the glucose 6-phosphatase. So it is a diagram for the glycogenolysis and the gluconeogenesis. So glycogen is converted into the glucose 6-phosphate with the help of the glycogen phosphorylase. Then this glucose 6-phosphate converted into the glucose with the help of enzyme glucose 6-phosphatase, and it is called as a glycogenolysis. Whereas in the gluconeogenesis, lactic acid, some amino acid and glycerol from lipids, which is known as non-carbohydrate source, enter into the gluconeogenesis to form the glucose 6-phosphate to glucose, which is known as gluconeogenesis. And the gluconeogenesis has the main four regulatory enzyme: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and the glucose 6-phosphatase. Some pathways in the gluconeogenesis, some steps in the gluconeogenesis are the reversible step of glycolysis. Next, glycogenolysis primarily occurs in the liver, and gluconeogenesis occurs in the liver and the kidney. Glycogenolysis occurs in the cytosol and it requires less ATP, whereas the gluconeogenesis occurs in the cytosol and the some steps in the mitochondria, and it requires around the 6 ATPs. So glycogen is break down by the glycogenolysis and it forms the glucose which enter into the glycolysis, and The product of the glycolysis is the pyruvate in the aerobic condition and the lactate in the anaerobic condition. Then it converts into the acetyl-CoA, which oxidizes into the TCA cycle to form ATP by the electron transport chain. Whereas some non-carbohydrate source like pyruvate, lactate and the glucogenic amino acid enter into the gluconeogenesis to form the glucose, and this glucose enter into the glycolysis to form the ATP. Whereas when the glucose form in the excess, it's also enter into the glycogenesis to form glycogen. So it is a difference and similarities between glycogenolysis and the gluconeogenesis. Thank you. If you like my videos, please like, comment and subscribe my channel and share with your friends.